like to introduce Ruth Menger, who's in the chemistry department at Colorado State University. Ruth?
Hi, thank you. I'm finding the correct screen here. Okay. So I would like to introduce some of my research that I've been doing for a paper-based analytical device on for the detection of PFAS in agricultural waters. First, what are PFAS? They are per and polyfluoroalkyl substances, and this name might sound familiar because there was a massive lawsuit about 20 years ago against DuPont. And also it's the topic of the film Dark Waters that came out a few years ago with Anne Hathaway and Mark Ruffalo. But these PFAS is a massive group of compounds, about over 4,000, and they all have this similar structure where they have a carbon backbone and lots of fluorines on them, and either a sulfonic acid group or a carboxylic acid group at the end. And this PFAS and PFOA are the two most common. And these compounds are found all over the place, like your Teflon pans, water, anything that's water or oil resistant. So those Teflon pans, any rain jackets, ski wax, are all have some sort of fluorinated compound in them. And during the manufacture and also disposal of these products, they can come from the industry and through the products and enter our environment, or even more directly from the industry. And they can enter our air, soil, and water and cause a variety of human exposure and negative health effects, like they can negatively affect our immune system, development in kids, and increased risk of cancer as well. And a big problem with these compounds is that they don't degrade, in that they bioaccumulate. So we're always going to have increasing concentrations of these around. It's a problem that's not really going to go away anytime soon. Here's a map of PFAS occurrence in the United States, specifically within drinking water sources. So the gray areas are areas that were tested, but none were detected. That doesn't mean there's none there. It just means that there's very small amounts that weren't able to be detected. And the blue areas do have occurrences of PFAS. And here in Colorado specifically, this blue area is down by the Air Force Base in Colorado Springs. Another big source of PFAS is firefighting foam. So anytime you have an airport or Air Force Base where you might have a fuel fire, they're going to have lots of quantities of this firefighting foam to put that out. And if a fire does pop up, they put it out. They will often have that firefighting foam will leach into the surrounding areas, into the groundwater and surface water, and then eventually our drinking water as well. Now, the EPA has set a health advisory limit of 70 parts per trillion for PFOA and or PFAS. This is a lifetime limit, but and it's also not regulated yet. There's a lot of public policy groups that are working on pushing that limit lower and actually making a regulated compound. But these are the kind of concentrations that we're aiming for, so pretty small. Specifically within agriculture, there's a lot of different ways people could be exposed. First, through the irrigation water, like I mentioned, if like an Air Force Base, the firefighting foam leaks into the ground and surface water, and then that water is used as irrigation water. Any biosolids or sludge from wastewater plants that is often used as fertilizers can have high quantities of PFAS. This then also goes into the plants and also the soil. They've been able to find 500 to 6,000 parts per billion, so orders of magnitude higher than the limit of PFAS in the soil. And the EPA has acknowledged this as a problem as well. They put out a call for proposals back in February to fund projects that deal with the occurrence, fate, and transport of PFAS, specifically within agricultural products. And part of all of this, to evaluate all of this, is we need a way to detect PFAS. And the common way of doing that, the EPA standard method, is through liquid chromatography mass spectrometry, or LCMS. It's a great method in that it's very sensitive and selective. It can achieve low limits of detection, and also you can pull out specific PFAS instead of the entire compound, or sorry, the entire compound class. However, oftentimes you need some sort of sample pretreatment, like solid phase extraction, to get rid of any other interferences. You need a centralized laboratory to run, and also trained personnel to run the instrument. It can be very expensive, $200 to $400 per sample, and also 40-minute runtime. None of these qualities really make detection of PFAS easy for any on-site detection. So if you had a farm and you wanted to evaluate the water that you're using for PFAS, Ideally, you'd have something that's, one, gives you results very quickly in the order of minutes. It's very cheap and also easy to use, doesn't use any external instrumentation. So that's what I've been working on. A microfluidic paper-based device, or micropad, is a nice option that my lab has been developing for a variety of analytes. And these are all based on paper. So paper is a super easily obtainable substrate. We are using Wattman filter paper because of the porosity of it. The fabrication is super easy. So here I have a scheme of generally how we make it. We use a commercially available wax printer to print a design on paper. And the ink has wax in it. So then when we set it on a hot plate and add some heat, the wax will go through the paper and create hydrophobic boundaries. Then we can add different reagents. So here we've added some reagents in these, the outer corners. We can add a sample, which then flows through the paper and is limited by the hydrophobic boundaries into the detection zones and changes colors. And we can tune those reactions for a variety of different analytes, for environmental contaminants, like I'm focusing on here, heavy metals, bacteria, biomarkers, viruses. There's really a whole multitude of papers and devices available for this, as you can see down here. Super easy to prototype. The detection is really fast, on the order of minutes, and typically instrument free as well. Or if there's an instrument involved, it can be your smartphone, which makes it really accessible. So specific for PFAS, I've been focusing on this methylene blue active substances assay, or I have been calling it MBAS, where this is the PFAS that we saw before, and when it comes in contact with methylene blue, which is it's a dye that starts out blue, they form an ion pair, and that ion pair is purple. So down here, I have a spot test, what we call it. This gray circle here is the wax boundary, and then I've added methylene blue to all the circles, and then PFAS to all the circles, and we get a nice purple color with a pretty decent calibration curve as well. Now, as you notice, this is down in ppm, and this is the big part of what I've been working on the project to lower the limits of detection. And for this analysis, I did scan the device and measure the color change, so the change in red, green, and blue color channels. We can get a visual limit of detection around 100, 250. The actual device in person, the colors are a little bit more distinct, and then calculated 10 ppm. So a big problem with this is that, like I said, I did use a scanner, or you'd need some kind of camera to analyze the color, and which just adds a few more steps, but we can both increase the sample capacity of this device to lower the limits of detection, and also reduce the amount, reduce that extra scanning step by making it into a distance-based device. So here the yellow is a wax in this case. I can very easily change the color. So I added the methylene blue to the straight channel, and then I can add PFAS down here, and then measure the distance of the purple color that is formed. So as you can see, as we add more PFAS, the distance that purple color travels is larger. So in this case, I also scanned the device and measured the change in color to get a nice calibration curve, but I can also either measure the purple color with a ruler, or a next iteration of the device actually has a little tick marks on the side here, which can then tell you, just read whatever tick mark that is, and that will tell you what concentration of PFAS you have. So unfortunately, I wasn't able to get a lot of lab time in due to our lab being shut down for COVID. So there's a lot of still work to be done here. First, the big thing is to optimize the device for lower limits of detection, to reduce it down to the part per billion level, ideally. Real water samples is also next. There's a lot of matrix effects. I've been focusing on drinking water because it's a relatively clean sample, we'll call it. But future work would involve expanding that to groundwater, surface water, also soil, and then blood would be a nice matrix to evaluate actual human exposure to it. And then for these devices, evaluating the stability for pH, time, and temperature is also important. With that, I'd like to acknowledge HICAHS, of course, for funding, and my two advisors, Dr. Chuck Henry and Dr. Thomas Borch, and then my two lab groups as well for all of their support, and I'd be happy to take any questions.
Great, thank you, Ruth. It's good to see the work that you're doing on low-cost, simple tests that are really going to make a difference in rural areas, or developing economies that have to deal with the budget's limitations they have. So nice job.
Yeah, they're useful all over the place.
Yeah. the question is, how optimistic are you that paper-based technologies can get down to a part per billion range?
Hi, thanks for the question. I'm pretty optimistic about it. For some of our other paper-based devices, I do also some work with heavy metals. We are able to get down to the part per billion range. And it's all about optimizing the concentrations of the reagents and also doing some device engineering to increase the sample capacity. We've seen is the biggest way we can do that. And based off my calculations, like what volume I can put on the device right now, and then how much I can increase it and therefore increase the sample volume, I'm pretty optimistic that I can get down to the part per billion range. I just have to actually do the experiments for that.
Great, thank you.